Anyways, as you can see here, it's a picture of a brain of one of my students that we took here at the Robarts. And you can see the stimulus going all around his visual fields. There's the left, the right, left, right, upper and lower. So you can see that whenever something on the left happens, there's activity here. And this is in the right cortex. So in the opposite cortex, you get activity in this area close to something called the calcarine sulcus. And you can also see that when everything's to the left but above, it's below the calcarine sulcus, and whenever something is from below, it's above the calcarine sulcus. So it's flipped not only from left to right, but up and down. So let's first see how this signal from the retina, which is where we left things off last week, ends up here in the calcarine sulcus. So you can see here images hitting the back of the eye, and the red lines appear when something is shining from here on the left, from the left, and here from the right. And again, you can see that things from the opposite visual field end up in the contralateral visual cortex. But to do that, one eye has to cross, and it crosses at the structure called the optic chiasm. So now, we saw in the eye that we had ganglion cells, and we had ganglion cells of different sizes. And those in the fovea were small, and those in the periphery were large. Those in the fovea first go to a place called the lateral geniculate nucleus. That's a part of the thalamus. Every one of your senses, touch, hearing, all go through the thalamus. And you can see that here, information goes to some of the layers of the thalamus. The ones from the periphery also go to the thalamus, but they also go to a structure called the superior colliculus. Now, the superior colliculus is important in generating something called the visual grasp reflex. So if somebody flashes his hand over in my periphery, I then look to see where the source of that stimulus is. And when I do so, then my fovea, which are these guys, are analyzing the scene. So the periphery doesn't have to be very accurate. It just notices that something's moving or changing, flashing. And then it directs the eye to turn, or the eye and head to turn, and points the fovea at it. And that then analyzes the scene. So the ones in the periphery are sort of where the object is, and the ones in the fovea are important in analyzing what the object is, because that's where you have your acuity. Remember, about half the cells from the optic nerve are coming from the fovea. Now you can see here that in this structure, lateral geniculate, you have red-colored layers and blue-colored layers. They're coming from different eyes. So the cells here just see what one eye sees or the other eye sees, not both. And also the ones from the periphery are going to these layers here, and they're called the magnocellular layers because the cells here are big. And these ones here go to the parvocellular layers, which, again, because the cells are small, are given the name parvocellular. Now, how do cells grow to the thalamus? So imagine that at infancy, the eye develops from part of the brain, and then these cells from the eye have to grow to the correct part of the brain. And the first part they have to go to is this lateral geniculate nucleus. And you've got a lot of ganglion cells, and how do they get to where they're supposed to go? Well, neighboring cells grow to neighboring locations, so that makes it easier for the brain then to figure out where they're coming from. And the way that happens is, first of all, in your socket, where the eyeball lies, that structure develops this chemical gradient. So you can see that on this side, you've got different shades of green, and this side you've got different shades of red. And that's supposed to symbolize a chemical gradient that's being developed in that socket. That chemical gradient is taken on by the retina and the ganglion cells that are located there. So you can see that this ganglion cells takes on this lightish green, mid-lightish green shade of green, and the other one, the same thing on the opposite side. So a similar then gradient develops in the lateral geniculate nucleus. So these cells here are light, these cells here are dark. This is green, this is red. That then attracts the ganglion cell. So you imagine this ganglion cell is sending out an axon, and it's traveling, and it's searching for the scent. So little sort of parts of it grow out, and then if it notices that gradient isn't very good, another part grows out, and whatever part sort of smells that gradient most, that's the direction that the cell moves in. And eventually it finds its way to the correct part of the lateral geniculate, in this case on the opposite side. Sometime later, the cells on the opposite side of the retina start growing, and they know not to cross because the scent over on this side is the scent they're looking for. And in this way, the lateral geniculate develops a sort of a visual representation of the retina. So cells over here are mapped to here, and you can see the picture of the retina on these layers. Now, one of the puzzles about the lateral geniculate is that we really don't know what it's doing. And that's important because every sensory modality goes through the thalamus, and we really don't know what it's doing for any of the senses. First of all, with respect to the lateral geniculate nucleus, we don't know why there's so many layers. It seems some of them are redundant. The other part that's interesting is most of the input to the lateral geniculate doesn't come from the eye, but from the brain itself. Okay. The third thing that's odd is that the receptive fields don't change. The receptive fields we saw in the retina on the ganglion cells are the same as you see here. So as I mentioned, this is an important mystery to try to figure out. Now, to where do these lateral geniculate nucleus cells project to? They project to the back of the brain. We saw that in the first slide, this structure called the calcarine sulcus. So that's this line located here. Now, you see that V1, the primary visual cortex, also called area 17, also called the striate cortex in some books. You see a little bit of it here from the side of the head. You have to look at the middle of the head. So imagine you take the brain and split it in half, and you look down that half that you split, and you see most of it, but not all of it, because much of it is hidden here in the sulcus, which is like a valley in the cortex. Now here we unfold that valley, so you can see here's the calcarine again. We've sort of unfolded all the valleys, all the sulci, and we can see clearly the whole shape of the V1. And we can see that if we shine a light over here, we activate something in that V1. And the reason here that we activated V1 is because here, this is the left visual field. So this little target here is, or right visual field. This is the right visual field. This is the left cortex. So we're contralateral. The other thing you can see is that here we're lighting something below the calcarine because the image is above where we're looking. And the same thing here. We're lighting up a different part of V1, but below the calcarine. The other thing to note is that this region here is at the back of the cortex, and this region here is slightly more forward in the cortex. So there's differences. The things around the fovea are mapped here, whereas the things in the periphery are mapped here. So fovea, periphery. Now, if we look even in more depth, we find that, again, this is the calcarine, this fold valley in the cortex. And this is made up of gray matter. Here's the surface. And below it is something called the white matter. So the gray matter is the important stuff. It's where the cells lie. And the cells are important because that's where the synapses occur. And as we'll see in a moment, that's where all your memories are stored. The white matter is also important because it has the fibers that all these cells send out. It's the wiring of the cortex. If we look at the gray matter in, again, higher magnification, we find that it has these layers. And the surface layer is called layer 1, and the deepest layer is called layer 6. In the middle here, in the case of the V1, your primary visual cortex, you have a very thick layer 4C. And that's where all the input comes in from the lateral geniculate nucleus. And that's why you have a very thick layer 4C, because of this massive input. Now, what kind of cells do you find there? Well, you find, again, first of all, cells very similar to what you found in the ganglion cells. They fire for something. This one here has an on-center, so it fires whenever there's a light in the center. You then also find above and below layer 4C these things called simple cells. Now you notice here that the receptive field is elongated, and it's elongated in this horizontal direction. And you can see when we play this, it doesn't fire for vertical lines, but it does so for horizontal lines. So lines that lie along this elongated edge. And you have simple cells that of all possible orientations. And so they're becoming more sensitive to lines. And then you find complex cells, which, like simple cells, are tuned to lines of a particular orientation. In this case, a horizontal line, but that horizontal line can be over a wide visual area, a large part of the retina. So it has this positional invariance. Now, how does this organization take place? How do you make these kinds of cells? First of all, to make a simple cell with an elongated receptive field, you take ganglion cells in your retina and combine them with LGN cells, and in turn send those to layer 4C cells. And finally, you combine them onto the simple cell. And here, to make a simple cell that's tuned to a horizontal line, you map cells that have receptive fields along a horizontal line. Then here we're making a complex cell that's sensitive to horizontal lines over its larger receptive field. Again, what we simply do is add all the input from these receptive fields, each of which is sensitive to a horizontal line. Now, complex cells get sometimes more complex. So this one here, you notice it fires a little bit here, a lot here. So this complex cell fires best for a line of a particular length, the length of its receptive field, not longer, not shorter. This one here is yet a different one. It fires whenever a line ends in its receptive field. So nothing there, and fires there. So why is this important? Why is this clinically relevant? I used to teach this to the second-year medical class as well, and they right about now, hands would pop up. Why are you teaching us this stuff? Why is this clinically important? Well, one of the reasons is that it helps us figure out how the brain is organized. So we go from ganglion cells to simple cells to complex cells. And we keep doing that. And eventually you have cells that recognize faces. So you go from very simple things like lines to corners to edges of all kinds. And in the end, you can recognize a face. And you do this by the process of having a cell that's very particular in what it looks for, and then generalizing that over a larger area of retina. So you could recognize a face if you look at it from a little bit from, not directly on the fovea, but a little to the edge, not too much to the edge, because receptive fields aren't very good. But you can imagine that this is the way the brain is organized. Now, the discovery, what's more interesting is the discovery of this simple and complex cells was done by a fellow called David Hubel together with a colleague called Thorsten Wiesel. David Hubel grew up in Windsor, not far from here. He then went to McGill and then won the Nobel Prize for his study of these kind of cells. And these cells help, he also showed that these cells are very important in developing something called amblyopia, which is visual blindness, even though there's nothing wrong with the eye that we'll look at in a moment. Okay. Now, what use of the complex cells? One example of that is from the description of what one patient observed. And in this case, the patient had a scotoma, or a blind spot, in his retina. And whenever a face would appear in that blind spot, he couldn't see the face. But if behind the face there was a striped wall, he could still see the stripes through that area. Okay? So the stripes were still visible in his blind spot. Now, how does that happen? How does the visual system see that? Well, the answer is that when a face appears there, there's no cells, because he's got the scotoma, that see that face. Okay? Either the scotoma is because all the ganglion cells in that region have been damaged, or because the cortical area that represents that area of the retina has been damaged. But out here, the end-stop cells can see that the line is ending. Another one over here can see the line is ending. And they then produce activity even though there's nothing in their own receptive fields. So they produce this perception of this line. Now, you might find that a little far-fetched. Okay? So what I'm next going to show you is that all of you are doing the same kind of filling in at this very moment. So, to do that, those of you that have these notes on a piece of paper can close your left eye, take your piece of paper, and move it forward, look at the X, and move the paper forward and backwards until you see the dot disappear. Those that have a screen can do the same thing by moving their face closer or farther to the screen until the dot disappears. Now, it should be about this length. Okay, the dot will disappear if you look at the X. Okay. It's best done with a piece of paper, but I'm trying to figure out. Yeah, right. Yeah, at about this distance, the dot disappears from my screen. So if you're looking at a screen, it's about a forearm's length. Okay, next, let's look at these other images. If you look at this line here, still staring at the X, the break in the line will disappear, and you'll see a continuous line going right through it. That's your end-stop cells doing that. Okay. Next, we see the face. And you see, if you look at the X again, the face will disappear, but lines will still appear behind it. And most amazingly, or more amazingly, is that when you see this blue spot, it will disappear, and you'll see yellow behind it. And finally, I think this is the most amazing, is that this pattern appears instead of the blue line, blue dot. Okay? So the brain is making things up. What you see isn't actually there. Now, your blind spot is not small. It's about the size of what the moon would look like in the sky, okay? Which is quite a big object. And if you close one eye, to think that there's a blank spot in your retina about the size of a moon right there, okay? And you don't notice it. It's kind of amazing. But it's the wonders of the brain that do that. Now, the visual cortex, by using these simple and complex cells, actually improve on what you see. So if you move your head different distances from these objects, you'll find that it's easier to tell that there's a break in these lines than it is to tell that there's a break in these dots. That this is one dot, over here is pretty well one single longer dot, and over here you can start seeing breaks in it. But it's easier to tell that that's happening from these lines. And this is happening because you're combining the information from lots of ganglion cells and in turn from lots of LGN cells and cortical cells and so forth. So developing a much better signal of this break from in the cortex. Okay, so the ganglion cells see dots, but the simple cells see lines. And by analyzing the segment, you get this hyperacuity, like an acuity that's better than that of the eye. And clinical letter charts use this hyperacuity to actually improve on what you can see. So when you get your eyes tested by an optometrist or an ophthalmologist, remember that what you're being tested for is not just your eye, but also how good your cortex is. So there are many more cells in the visual cortex than there are in the LGN or the eye. You get an explosion of connections. And what's happening is that, for example, here, this one cell here in the middle of your LGN, and you can think of that one cell as being one cell in your retina, is being shared by lots of simple cells. And you can see that that's contributing to these simple cells, all of which sense whether the line is horizontal, vertical, or at this halfway point. But in actuality, you have simple cells that measure very fine differences in angle. So you've got this simple cell A is actually sending information to hundreds and perhaps thousands of other simple cells. So you can see that's the region for the explosion. It's got to share this information to many more cells. Now, what does it look like when you look at the cortex in greater detail? Well, as I mentioned, at the back part of the visual cortex, there's a fovea, and then there's the surround. And you see these little cubes, or not cubes, but squares. Anatomically, there's no such thing as these squares, but functionally there is. And these are called hypercolumns. And you can see here that this is what a picture is in real life. There's where the nose is. And because of the expansion, there's much more representation of the signal from the fovea. And this is what let's say you're looking at here. The nose has a much bigger representation, okay, starting at the retina and going all the way to the cortex. So there's a distortion of the picture. The other thing that happens is you get all this input being organized. Now over here, that's the input coming in from the LGN. This is your layer 4C. These cells here, when they come in, they have these types of receptive fields, on, center, off, surround, or the opposite. And they're all monocular. They're just receiving information from one eye or the other eye, but not both. When we go up or down to these other cells, they're the ones that see lines of a particular orientation or complex cells. We start finding cells here that see from both eyes, or are called binocular. And these binocular cells are what allow you to see depth, things in 3 dimensions. When you look at a microscope with through a binocular microscope, you can see the image in 3 dimensions. That's because of these cells. So each hypercolumn extracts the following features. First of all, it extracts this stereopsis, or depth information, and it does so in these areas here. So these areas here and this area here, at the edges between the 2 columns, hypercolumns, is where you find your simple and complex cells receiving information from both eyes. The ones here in layer 4C are just receiving information from one eye. The other thing that you get is down the center of each one of these hypercolumns, you have cells that respond to color. And this is where those double opponent cells that we talked about in last week's lecture come from. And then around these columns, or blobs, as I forgot to mention, because that's what they looked like when people did the anatomy, you find these spokes of a wheel. Okay, and in each spoke, you find cells with a similar orientation. And so, as this line turns round and round, these spokes light up in series. Okay, neighboring orientations are mapped into neighboring spokes. And so the reason for that, again, cells that are like each other like to be near each other, and they do that because the connections that you need to form these types of cells are shorter than. Okay, so at birth, in these higher layers and lower layers, this is layer 2, you find that initially all the cells are binocular, so they're receiving information from both eyes. The red is coming from one eye, the green things are coming from the other eye, the left eye. Now, if the eye develops normally, then these cells become tuned to one eye or the other eye, preferentially. So here in the middle, this one here will be just receiving, there will be a simple cell just receiving information from the right eye, this one here from the left eye. These ones at the edges, as we mentioned before, start receiving information from both eyes, the binocular cells. And over here in layer 4, that we can't see at the moment, they're all monocular. Now, if something goes wrong in one eye or the other eye, the one that's the better eye starts forming stronger connections to that part of the cortex. So here we have something wrong with the right eye. It's weaker in some ways, either because there's some opaqueness on the cornea or some other defect. As a consequence, signals from the left eye start taking over that part of the cortex. And if the newborn, that this problem persists, this eye will completely take over the cortex. And if, let's say, at the age of 3, that defect is fixed so that both eyes see equally well, that defect will remain. The child will remain cortically blind from that eye. Okay? So it's important to if there is some defect, either something in the cornea, to fix it early. And that's because the cortex, the visual cortex, has this thing called the critical period, and it has an early sensitivity. And you can imagine that There's competitions occurring for representation in the cortex. Whatever has the strongest input will take over from the cells that have a weaker input, for the synapses that have weaker input. Now, studies have been done in kittens, and they found that if they raised kittens in the dark, they could prolong this critical period. Now, that doesn't mean that the visual cortex is totally without any plasticity. It can still be adapted later on, but not in terms of regaining one eye or the other eye if it has lost it. How does that happen? Well, plasticity depends on synchronous activity coming in. And the key is this blue thing here called the NMDA receptor. And it opens only when the cell is very strongly depolarized. So if two of these synapses fire at the same time, they'll produce a stronger depolarization than if only one fires. So the sentence to take home is that if cells fire together, they'll wire together. And while this might seem like a simple concept, it is the basis of all the plasticity that occurs in your brain. And this is the basis through which everything that you learn forms changes in your brain and is how you remember things. So what are the steps? So first of all, you have a synchronous input. So the cell depolarizes because these two, the green ones, fired. That opens up the channels, that strong depolarization, just on these guys, because they're the ones that fired, and they inject calcium into the cell. That in turn causes the release of NGF, a neural growth factor. And that NGF just hits the cells whose channels were opened. Okay? And that in turn causes those synapses to get stronger by growing bigger, making a tighter connection, whereas the other ones that didn't fire recede. So synapses are strengthened and the other ones weakened. And this is how memories are formed and memories are forgotten. So you can see here, the green ones are firing simultaneously. The red ones are firing asynchronously. So synchronous versus asynchronous. So the green ones, because they fired together or synchronously, form these strong connections. These ones, because they fired at different times, got retracted. Now, in this case, all of them are firing at the same time, both left and right eye, and both eyes maintain their strong connections. So you can imagine that in your visual field, this line of this auto orientation fires, and it's got to activate cells in the right eye, left eye, and the right eye. And it's got to get to the cortex and fire this simple cell. And it's got to find this one simple cell with connections from both eyes. Okay? But we remember that at birth, many cells have binocular connections. Okay? So what the brain is doing is pruning those connections that aren't ones coming from this same line at a particular orientation. So only those that are coming from the same visual field and the same orientation will keep their connections. So this this process, this critical period of producing that can produce amblyopia, has an ulterior motive. It has a purpose. It's trying to tune your visual system to all these little fine things like to produce hyperacuity because it's sensitive to these lines or particular orientations. Okay. Now, sometimes eyes, as in the previous case, have something wrong with one eye or the other eye. But sometimes eyes point in different directions. You have strabismus or cross-eyedness. If you have that, then you never get the same image on the two eyes, okay? One eye will see one thing, and the other eye will see another thing. And so you could never develop these simple cells that see the same thing at the same time. And when you do that in your critical period, you lose these binocular cells. So this is the normal binocular cells above and below layer 4C. This is what you'll get, monocular cells instead of those binocular cells. And as a consequence, you will never be able to see things in depth. Okay? So again, when a child is born that's cross-eyedness, it's important to get the two eyes looking at in the same direction at the same time. Okay, let's see. If you want to test your abilities to see in stereo, try this. You can either try it on the screen or wherever you're looking at. Cross your eyes. If you cross your eyes, so instead of 2 squares with a flower in it, you see 3 squares with a flower in it. The one in the center will do amazing things. If you look at the one in the center and move your head, you'll find that the end, the flower that's part of the flower that's closest to you, moves whenever you move your head. If you move your head back and forth, the one in the center will be in depth. And what you're seeing there is what these binocular cells are seeing. Okay, so I'm doing this now. So for a screen, this is about the distance you have to be able. You try to look at something a little bit in front of the screen to get that effect. And if you want to get an effect on the screen in the back of the room, again, look at something a little in front of the screen in the back of the room. Okay, if you do the same thing here, you'll find in the middle one of the 3 that this line is tipping in and out of the screen. Okay. And so that's because different images are seen by the 2 eyes. So you look at here in the middle, some cells see this, so there's no disparity. They see the same thing. This is what the one eye sees, this is the right eye, this is the left eye sees. So over here they say the same thing. But as you go further up, different simple cells will start seeing this disparity. At this simple cell will see the thing on the left eye a little over on the retina than this thing from the right eye. And the opposite disparity over here. And so here we see one of those simple cells, you can see, or no, not one, first of all, the different disparities that the eye will see. So if the eye can see something closer or farther, you get, this is the view of the left eye, this is the view of the right eye, this is what the 2 eyes see. Okay? And we'll find cells that like particular disparity. So this is a cell here, a firing of one of these simple cells in the cortex. And This is what the eyes see, and this is what you can imagine the simple cell sees, although it's actually a line that it prefers. And you can see as I move the object closer and closer, the firing rate starts getting higher, and then higher still, and then dies away over here. So this cell prefers this particular disparity, just a little bit off the retina fovea in both eyes, but in this particular direction. And it's whenever it fires, the brain somehow recognizes that it's closer, a cell that's closer to you. Okay, we're almost done, and I'm on time. I promised that these lectures would be less than an hour, and we're doing fine. So, in summary then, the eye flips the image that it sees, okay? It then distorts the image that it sees by sending more information out from the fovea than the rest of the retina. The image then is combined in the visual cortex from the two eyes, and by comparing the image from the two eyes, you then get this sense of depth. Now, the cells in the cortex do more than just give you depth. We'll see that they also produce a sense of motion. Some of the some of the complex cells are motion sensitive as well. Some of the cortical cells produce color, the double opponent cells, and all of them give you the sense of where an edge is, so accentuate edges. And that's it. Thank you very much.